Hi everyone, it's Dr. LeBlanc again. This is part 3 of chapter 12, our final part, discussing how we can go from chromatin to organized chromosomes. Okay. In the nucleus of a cell, there are a lot of structures that are important to maintain the structural integrity of the genome. Okay. Remember that a nucleus has a double membrane structure. Okay, it has nuclear pores. Underneath the membrane on the inside of the nucleus is a nuclear matrix and nuclear lamina, and then in the middle of the nucleus is a nuclear matrix, which is comprised of lots of different types of proteins. There's internal nuclear matrix, there are other proteins that are bound to the nuclear matrix, and in the middle of this is all the DNA that makes up the genome. So in order to get further compaction of the chromosome, further compaction of that 30 nanometer fiber that contains the core histones plus H1, the chromatin has to be attached to the nuclear matrix. This is an actual picture of a nucleus from which all the chromatin has been removed. So this is a nucleus without any DNA in it at all. And you can see that it is not just an empty sac with nothing in it, but it has lots and lots of proteins, lots and lots of these nuclear matrix proteins in there. Okay, one of the basic structures that the chromatin takes up inside the nucleus is called a radial loop domain. Okay, there are approximately 25 to 250 base pairs, quite a great range of DNA, sizes of DNA, within these radial loop domains. The matrix attachment regions are specific regions of the DNA that bind to proteins that attach to the matrix. They're also called scaffold attachment regions. The MARS, the matrix attachment regions, are anchored to the nuclear matrix, thus creating these radial loops. And notice that the active genes are almost always contained within these loops, not in the parts of the DNA that are attached to the matrix. And remember that this is a 30 nanometer fiber. It's not just the double helix of DNA, but it's the DNA wrapped around all the nucleosomes all the way around. Okay, this is a nice micrograph showing the nuclear matrix. Some of the regions that have less proteins in them are pointed out, fewer proteins in them. A chromosome has regions of chromatin that are very tightly packaged at the telomeres and the centromere, again, and at the telomere, and then regions of chromatin that is more loosely organized. Those two designations are referred to as heterochromatin and euchromatin. Heterochromatin is the very tightly packaged DNA at the telomeres and centromeres, and the euchromatin is the DNA that's not quite so tightly packed. The euchromatin has the 30 nanometer fiber anchored in radial loops, whereas the centromere and the telomeres are heterochromatin, where they are a greater compaction of the radial loops. So if you look at a chromosome, and if you go all the way back to its basic structure, you can step through the hierarchy of organization. So if you start with a double helix of DNA, here's a nice picture of just plain old naked DNA right here. It has a diameter of approximately 2 nanometers. When you wrap it around the core histones, it's 11 nanometers. Okay, it should not have these histone H1s here. This is a mistake. It should not have these. Okay, when you add the histone H1, you can form the zigzag or solenoid structure that makes the 30 nanometer fiber. Okay? And this is really the basic 30 nanometer fiber of chromatin. Okay? When you anchor this 30 nanometer fiber to the nuclear matrix via the radial loops, and the structure of the radial loops, you get a 300 nanometer fiber. Okay, so here's our protein scaffold and the radial loops, and notice their varying lengths. Okay, and then further compaction of the radial loops creates a 700 nanometer fiber. And finally, when you have a bivalent metaphase chromosome, the width of that is approximately 1400 nanometers. Here's a metaphase chromosome with the scaffold, and a lot of the proteins extracted leaves you this pattern of swirly DNA right here. There are two multi-protein complexes that help form and organize metaphase chromosomes: condensin, which plays a critical role in chromosome condensation, and cohesin, which plays a critical role in sister chromatid alignment. Okay, a condensin molecule will help stabilize these radial loops. Condensin is a complex of proteins, not just one single polypeptide, but several polypeptides together. And if you want to learn some more about this, you can go to this link right here. We all love these question marks in science. Cohesin is this little red dot of a protein here that's holding two sister chromatids together. Here's a centromere region. Here's a chromatid. Here's its other sister chromatid. This whole thing is called a chromosome. During metaphase of mitosis, this chromosome compacts, but eventually these two sister chromatids have to go their separate ways. One set to one cell, daughter cell, and one to another daughter cell. So these cohesin molecules have to be released by the chromosome, and eventually the two sister chromatids can separate during mitosis. If you don't remember the phases of the cell cycle, this is a good time to review them. At the end of S, or the synthesis phase, during the G2 phase, the two sister chromatids are held together by cohesin. At the beginning of prophase, they condense even further. In the middle of prophase, the cohesin is released and the sister chromatids are freed. And finally, at anaphase, the cohesin that remained at the centromere is also released and the sister chromatids can separate. That's it for Chapter 12. Thanks for listening.